So this is my mentor, Andrea, and she's from Bogota, Colombia. She graduated from electrical engineering and psychology at the University of New Orleans. In the present, she's a PhD student in computer science at Tulane University, and she specializes in applying artificial intelligence to marketing. She currently lives in New Orleans, and she likes to run and loves her dog Mila. So for my project, we created a code that locates the origin of replication in a strand of bacterial DNA, specifically Vibrio cholerae and E. coli. So it's important to understand how replication works and what causes it to malfunction, because an error can lead to various diseases such as cancer, and we can prevent this if we know how replication works. So in order to understand the entire process, we must first know where to look for the origins of replication. So for this reason, to do this, we used Coursera, we used Codecademy to learn about biology and programming, and we used Python and GitHub to save all of our work. So what we did first is pattern count is the most frequent pattern of a certain length of nucleotides. It's also called a chimer, and it's found in a text of a certain section of DNA. So what we did first is we used a threemer, which is 3 nucleotides, and as you see in this picture, it says CGA on the first line. That's a threemer. We ran a window through this text, and every pattern that came up, the code would sort of place that pattern into a list, and for every pattern that came up again, a number would come up next to it. So over here, ATA comes up 3 times, so next to ATA would say 3. And then we went on to a little bit further, and we said that we used a pattern of 9 nucleotides, and that's what research suggests is the most common length of an origin of replication. So in a text length of 500 nucleotides, we found that the most frequent 9mers were these, and each of them occurred 3 or more times, and these 2 were complements of each other. And we concluded that because these 2 strands occurred the most, they were most likely the origins of replication. So then we went to another type of bacteria, and we found that there were many regions and many possibilities that could be origins of replication. So we had to think of another method. So what we did was, we did a little bit of researching about this, and what we did was we figured out that there are 2 orders of replication, a reverse strand and a forward strand, and a reverse strand and a complement strand. And in the forward half strand, which is in the forward half strand, meaning the one, but in the forward half strand, there is less C's, like the ATCG. There are less C's in the forward half strand. And we concluded that the area where replication ends is the area with the least amount of C's. And if you look over here, this is the ending. And if you look across from it, you can find the origin. So this is how we concluded the origin when there were many, many different possibilities of origins. We also did another thing where we found this one is very simple, but it basically runs a window size of 4, so 4 nucleotides or 4 letters, and it starts from this index, and it goes up to the 4th letter. And in the 4th, it wants to see how many A's there are in that certain window. So what this does is, in this first index, there are 4 A's, and from the second A until the first G, there are 3 A's. This is a more simpler code that we did. So we used this in a text of 4 million characters and a window size of 500, and we looked for the characters C and G. So we computed each position of C and G, and we looked for the maximum amount of C's. And once we found the maximum, we looked across and we found the minimum. And that would be the origin of replication. So my demonstration demonstrates how to find the most frequent pattern in a code. It's a simpler one, and it also it computes the reverse complement of a given strand of DNA.